It sounds like there's a lot of people having a misunderstanding on the lab. On the lab write-up, I only expect you guys to talk about your day, because there's no way that you guys will have an understanding what happened the rest of the weeks, the rest of the week. Now, when you start talking about this, there's some data that you took, and then there's some pieces of information that you had. We talked about it when we were out there. Okay, so from a management sense, there are primarily three major concerns. The first concern is the plants that you're growing. This is a farm. Okay, what are the other two concerns when you're thinking about managing at Dillman Hill? Contamination. Contamination is one of them. Okay, so that's two. That's actually three. What's the other one? It's about soils. Not that that was a big clue. Water, drainage, but why in particular? Water holding capacity, why in particular though? What's the soil that you guys are dealing with there? The Hudson silty clay loam, it's eroded, you're looking at clayey soils. So you have three big management concerns. Plant material, soil clay content, and then this issue of contamination. Okay, now you have a number of pieces of information. You have mulch information, you have penetrometer information, you have bulk density information, you have temperature information, and you have water information. Now, the other thing is, you are not expected to write up anything about part B, which is the nutrient analysis component. That's for another lab. Okay? So you're only operating on those pieces of information you have, one other thing, and I think this throws people off a lot, is that we've already been making management decisions there. Okay, and our management concerns have been those big three that we just talked about: plant selection, clay subsoils, and contamination. Okay, what are the things that are being done on site to deal with that? Now, this is an active site, so there should be a lot of clues to you right there. What are we doing? What are they doing on site right now? Cover crops, raised beds, keeping the soil covered at all times, plant selection rotation, and there's one more and I can't remember it. Irrigation is part of it. It has to do with raised beds. Oh, nutrient management is the organic material coming in. Okay, so they've already made some of these decisions. I want you to take a look at what they're doing, or maybe you have some other ideas about how to deal with these problems. Okay, does that make sense? Yes, kind of ish. And I do not expect you guys to look at all 4 days. There is no way, if you were in Monday's group, that you know what's going on in Thursday's group. Look at your day. Look at those groups. Okay? Cool beans? Questions? Go.
Can we have it due at midnight instead of 5?
As long as it's in by Friday, give me the timestamp, it's fine. All right, we good? All right, so let's continue on pH, and this is actually the next lecture, but what I want to do is I want to take a step back for a little bit, because I realize that there's some questions about what acids and bases are and things like that. So I want to take a step back, okay, and I want to talk about this slide right here. Now, you've seen this slide before, and you're going to see it again, okay? What I want you guys to think about, we're talking about pH. What I want you guys to think about is I want you to think about what's going on in this soil solution and what's going on these exchange sites. Okay? The exchange sites are obviously controlled by how much CEC there is, cation exchange capacity. But the exchange sites, the cation exchange capacity has a certain amount of ions that are on those exchange sites. Now, if they're bases, we start calling it base saturation. The measure of how many bases are on that cation exchange capacity is base saturation. But all of these sites are occupied. Now, if they're not occupied by bases, they're going to be occupied by something else. Protons, because we're talking about pH, protons are one of the most important ones that are there. Aluminum's there as well. Other cations that are not considered a base. Aluminum is that one. Okay, so the exchange complexes are going to be dominated by stuff. Now, the reality is that these are in equilibrium. As I change the solution, the concentration of the solution, I add protons in here, I add bases in here, I subtract bases from here. I have leaching processes. As this solution changes, the exchange onto the cation exchange, the transfer of these ions that are in solution are going to change the ions that are on these cation exchange sites. So if I start increasing the proton concentration over here, some of those protons are going to start knocking the bases off and basically put these two pools into equilibrium. Right? Does that make sense? So I can fertilize. I start adding calcium. What's going to happen? Well, I'm going to increase the calcium concentration in the soil solution, which means that more calcium is going to start bouncing onto the cation exchange sites and knocking things off, putting this pool and this pool in equilibrium. Does that make sense? Okay. That was point number one. The other point is this issue of what is an acid versus what is a base. Now, we call we call this a base cation. It's not truly a base. Do you guys from chemistry, you guys remember what acid bases are? Acid bases are pairs. Okay, so here's an acid. We'll use sulfuric acid, for example. H2SO4. This is the acid component, and this is the base. Now, there are strong acids and strong bases, and there's weak acids and weak bases. The stronger the acid, the more there's going to be dissociation of the proton from the base. Does that make sense? Yes? The weaker the acid, the less dissociation of the proton from the base. Now, the other interesting thing here is as these protons leave the base actually gets stronger. Okay, so right now, this is a very strong acid. The first proton is definitely going to dissociate. So what I'm going to end up with is this. Okay? Now, by this proton dissociating, this base is getting stronger. It has a stronger charge now. Here it's bounced. Right? Now, if this proton leaves, this base gets even stronger. So in essence, the story is this base can potentially neutralize these acids. Does that make sense? Now, why do we call them base cations? Well, let's go back to that soil solution. Let's do it this way, actually. Let's use it over here. Here's my colloid, right? Okay, and on this colloid, along here, there are going to be bases and protons attached to the cation exchange sites, right? Now, in the soil solution, I'm going to have a relative proportion of protons to everything else, right? These are the bases, or just call them non-proton cations. Okay, we'll leave aluminum out of the story right now. Okay, so we'll just call these cations. Now, with that story that we talked about up there, this solution, the ions that are in this solution are going to be in equilibrium with these guys, right? Make sense? Now, what happens when I start adding protons? I lower the acidity, and I start adding protons to this. The relative concentration of these guys are going to go up, right, compared to these. But that means that more of these protons are going to start exchanging over there, right? Which means more of these cations are going to be coming off. And in essence, these cations are neutralizing the proton. They're acting like bases. And that's why we call them base cations. Does that make sense to everybody? Questions on that? Go. Because what's happening is the proton is going onto this exchange site and it's now no longer in solution. Okay, now, there's another step here, and I want to hold off on that. But the pH that you measure, the pH is associated strictly with the soil solution. It turns out that there are two other pools of pH, and they're all in balance with each other. But this is what we measure, and this is actually the one that determines the solubility of stuff. But there is also a pH that's associated with this cation exchange component, and there's also a pH that's associated with the structure of the colloids, of the material, the parent material that's in the soil. But right now, this is what we're talking about. Go.
So you say that the proton increase off of base cation ions, and you said that neutralizes the H plus.
Right, because what happens is,
The plus charge neutralizes the plus charge.
because what's happening is this proton, so imagine there's a potassium here, right? This proton that I just added in is going to knock that potassium off. The proton's here, the potassium is now in solution. So in essence, this proton has been neutralized from this acid pool, from this active pool of acidity.
Okay, well, wouldn't that proton react with an anion and then?
This proton can react with other things, but what's going to happen is this, I'm going to have an exchange here, back and forth, until this pool is equalized with this pool. Okay, does that make sense? Now, I can do this the other way. I could start adding potassium into this system. What's going to happen to the pH of this pool if I start adding potassium? Initially, what? Okay, go ahead, answer. Oh, it's a question? Okay, hold on. What's going to happen? Well, I'm going to increase this component. So in essence, I'm going to be lowering this relative. In essence, I'm going to be lowering the pH. But what's going to happen is those potassiums are going to start reacting this way, and this proton is going to start coming out. So I'm going to have an initial dip in my, a raise in my pH, a dip in my concentration of protons. So my pH is going to go up, but then it's going to go back down a little bit. Not as much as it was before, because those potassium ions are going to knock those protons back off into the soil solution. Does that make sense? So it's neutralizing it temporarily, but it's certainly neutralizing it out of this soil solution. And that's why we call it a base. Question?
So potassium has a larger ionic radius than hydrogen.
Yes.
But that doesn't matter. It's just the mass action.
Yeah. Okay. So the question was, well, because potassium has a larger ion radius than protons than a proton does, certainly you would expect if it was a one-on-one exchange, the potassium would definitely bounce the proton off, and the proton would go into soil solution. So potassium would go on to cation exchange sites. But we're really talking about mass action. It's not like there's one potassium and one proton. We're talking about thousands of them in the soil solution, right? And so even though one-on-one, the potassiums are definitely going to be exchanging, there's just so many of them that there's sort of an equilibrium in solution versus the cation exchange sites. Go.
So I thought pH was a gauge just of protons in solution.
It is, but by changing this ratio, I'm moving things that way and back and forth.
But if you increase the cations in solution, you're not really affecting the proton immediately.
No, you're not. Yeah, so
the pH would stay the same.
the pH would,
But then it would knock off protons, which would make it more acidic.
it would make it more acidic.
So by adding base cations to solution, you make the solution more acidic.
No. By adding base cations, you're actually going to raise the pH. Because what's happening is there's an when you're measuring your pH with these tests, you're basically measuring you're measuring everything. You're basically measuring the whole charge. Okay? And so you're also measuring this, but this is in relative to this. So while the change here you haven't changed the absolute number of protons, you've changed the relative measurement of that charge. And so the pH will change in that sense. Because what's going to happen is I'm going to get this kind of exchange. Now, generally when we do pHs, unless we're doing some sort of treatment for it, we can basically take a pH measurement and we can almost get it instantaneously. But next week or 2 weeks from now, when you guys start doing treatments, you're going to be adding things to it. And what you're going to have to do is you're going to have to let it sit there for a while so that this equilibrium can in fact establish itself. And then you get the pH of that. And the other important component here is you're measuring the pH of the soil solution, not the total acidity of the system. Does that make sense? Kind of? All right. It's this issue of equilibrium and the relative concentration here driving this equilibrium. Okay, do we feel fairly comfortable? Go.
So this is not a static process, and this is constantly happening?
This is constantly happening. This is not a static process. Okay, and even when nothing's been added, not that this is a real world scenario, but even when nothing is happening here, these things are still exchanging. I don't change anything over, I'm not adding calcium, I'm not adding protons, nothing. We put this in a jar, okay? Close it off, it's a closed system, all right? We've got clay on one side, we have solution on the other. The reality is these ions are constantly exchanging. Okay, but the reality is they all sort of bookend, or they all sort of equilibrate around, or steady state around a certain area of relationship of charge over here versus charge over here. Does that make sense? Go.
I'm a little confused about the cause and effect of neutralization. So if you add H plus into the solution that bumps out cations
Right.
from the colloids, you say that is the cation neutralizing the H plus, but the cations aren't doing anything. It's just an effect of H plus.
That's true, and that's why it's not truly a base, but it's acting like a base. Because imagine this, if this cation exchange site was totally dominated by protons, and I added more protons out here, yes, the protons are going to knock the protons off, but a proton's going to be coming out. So I'm not changing the pH. But on the other hand, if this was a potassium, I'm effectively, because of this exchange, I'm effectively neutralizing that proton because it's now here, and I now have a potassium out there. So it's effectively neutralizing it. It's not like a base where it's actually binding it. And that's truly a base up there. This is what we call a base cation. Okay, do we feel comfortable with this? Because we're going to talk about this more, but I want everybody to be somewhat understanding what we're talking about. Okay. The other thing that I think we ended up fairly confused is this from last lecture. Okay, and this is aluminum is a source of protons of acidity into the soil solution. And I think the confusion was this idea that, well, the protons are going on and exchanging. No, it's not really what's happening. What I want you to think of this is a weathering process. The aluminum, I drew these lines in here to give you a better idea. The aluminum is literally part of the structure. It's part of the colloid. The protons go in there and they start dissolving it, chemically weathering it. Okay? If they chemically weather that the aluminum can go into solution. Now, granted, the protons could potentially go on to cation exchange sites, but they're not going to become part of the structure. They've literally dissolved the structure. Okay, that's step one. Step two is the aluminum, now in solution, will react with the water. And it will hydrolyze that water. It will break it apart. Okay, and that's this sequence right here. Here's the aluminum. It's reacting with that water. Okay, and it's going to make an AlOH plus 2. In the process, though, it releases a proton, effectively acidifying the system. Does that make sense? Now, this process can continue depending upon the different pHs. It can continue. This aluminum AlOH plus 2 will react with another water, releasing another proton and making LaOH 2 plus 1. This can continue reacting with another water and releasing another proton, effectively making gibbsite, AlOH 3. It's no longer charged, and basically it's taking up those hydroxides. It turns out that this is not a static system, and this is basically pH dependent. At low pHs, or as to say, if you start at a high pH and you start adding acidity, you're actually going to drive this way. Okay, because the AlOH plus 3, this OH will in fact dissociate as I start adding protons. If I start adding protons, it's going to react with that hydroxide, make a water, and I'm going to end up with this. And this is effectively a buffering reaction, and we're going to talk about that later today. Question? Gibbsite is a specific type of mineral. Gibbsite is like goethite is iron hydroxide, iron oxide. Gibbsite is an aluminum. Okay, this is effectively the building blocks for the octahedral sheet of a colloid. Okay, of a phyllosilicate colloid. Cool beans. All right. All right. I'm taking a deep breath this time. All right. So we have an understanding of what pH is. So let's ask the next question. What's going on in the environment? How is that pH? Where is that pH? And what we're going to talk about today is basically the pools of acidity. And I sort of introduced them over there. There's basically 3 pools of acidity. Okay? The other thing we're going to be starting to talk about today is buffering, and that's where we start talking about aluminum and stuff. Okay? But let's start with these 3 pools. And the best way to look at it is to sort of visualize it. Okay, here's that colloid again. Okay, here's the soil solution. Okay, this pool is the active pool of acidity, the pool that's in the soil solution. Okay, the second pool is the pool that's basically associated with those cation exchange sites. We call it the salt replaceable pool. Okay, because salts you can replace it. Right, this is the exchange pool. Now we also have what we call the residual pool. Okay, and that's the pool of acidity that's actually associated with the structure of the material, i.e., if this stuff weathered, what would be coming out of it. We call this the active pool because it is in fact very active. This pool, salt replaceable, this is the pool that's associated with the cation exchange sites. This pool is the pool that's slow to release because you literally have to dissolve the material to get it. Okay, does pretty good straight? Question?
What was the third pool called?
The residual. This is called salt replaceable. Sometimes it's called exchangeable. I actually usually call it exchangeable because it's the cation exchange sites. But both terms are acceptable. Okay, let's talk about the active pool. This is the ion concentration, the proton ion concentration that's in the soil solution. This is the pool that you measure. Okay? It's an extremely important pool because this is the pool that determines the solubility of substances in the soil. Not the pools, not the structural pool, and not the exchangeable pool. Okay? Again, this is what you actually measure. The next pool is the exchangeable pool, and this is the pool that's associated with those exchange sites. So imagine I have aluminum and a proton on those exchange sites. If I put potassium, there should be a plus there. If I put 4 plus 1 potassiums in this system, they potentially will exchange with these on the cation exchange sites, putting the aluminum and proton into soil solution. Direct effect is I've now acidified by plus 1, but also because I put this aluminum in there, I potentially will be hydrolyzing water. Okay, that's that pool. Does that make sense? Questions about this one? This is cation exchange capacity. This is the protons and the aluminum that are on the cation exchange capacity, are on the cation exchange sites. The next pool is the residual pool. This is non-exchangeable stuff. This is literally the protons dissolving the clay or the organic material. It's literally taking the aluminum, magnesium, iron, taking it out of the structure by dissolving it. You can see that the clay micelle in this example gets smaller. The protons are still in solution, but I now have these guys that were once part of the structure. I now have them in the soil solution. Okay, they are now part of the active soil solution. These guys are not going to contribute to pH other than acting as bases. This guy potentially will hydrolyze water and actively increase in the pH of the soil solution or the active pool. Okay? Lucas.
If that is, if from that
No. See, in this case, they're not exchanging. They're actually, they're weathering. They're chemically weathering this structure. They're dissolving it. So in this, I tried to make this drawing so that it looked like these guys are actually part of the structure, and they're not at the edge. They're not on the cation exchange sites. They're inside the structure. Okay, the proton then dissolves it. I mean, this material, you throw it into acid, it's going to weather away. It's going to dissolve, right? And as it dissolves, these things get released. Okay, the protons, potentially they'll go on the exchange sites, but the protons really are still in solution. But what happens is the clay starts this big, and it's now this big, and these things are now in solution. Does that make sense? So it's different than this scenario, where these are on the exchange sites, and this is exchanging for it. In this scenario, the protons are literally dissolving it. Okay? Questions about that? No? Lucas, does that answer the question? Okay. All right. This brings us back to the aluminum one. And I threw it in here again just so you guys can see. I want you to realize what we were talking about. These protons are literally taking this aluminum out of the structure. It's not like this aluminum's on the exchange sites. It's literally taking it out of the structure. Now, those protons can, in fact, bind, and there's still aluminum associated with this structure in other parts. But the aluminum now is in soil solution, which then can hydrolyze water and turn it into acidity. But this is now into the soil solution. Go.
Does this make the soils more acidic? Because in the beginning, before the aluminum was dissolved off, there's three H plus ions in the soil, and then when the aluminum is out, it's going to have the AlOH3 with three H plus.
In general, weathering processes over long periods of time will acidify systems. Now, that's initial stages, that's not necessarily true, because if this was a material that had lots of bases in it, by putting acid in and weathering away, I'm releasing all those bases into soil solution. But sooner or later, it might take 100 years, it might take 1,000 years, but sooner or later, those bases are either going to be taken up into plants, which are then lost from the system, or those bases are leached from the system. Yes, some of those bases can hang on in the cation exchange capacity, but as long as I keep acidifying a system through rainwater or something like that, the system will ultimately drive towards a low pH soil. Does that make sense? It may take a while, but it will happen. All right, bring it all back together. Here's that active pool. This is the pool that we measure, and this is a very important pool because this is the pool that controls the solubility of stuff. We have the exchangeable pool or the salt replaceable, okay, and this is in essence cation exchange capacity, the back and forth of those exchange, and then finally we have the residual or structural. Oh, I shouldn't call it structural. Sometimes it's called structural as well, okay, and that's literally the pool of acidity that's associated with the structure of the soil. You feel comfortable with that? Questions? Not a lot of glazed looks at the moment. Maybe it's just sleepy looks. Go. The plants are basically taking stuff up from this pool. Okay? And so the proton pumps and stuff like that are pushing stuff into here. Now, this pool is actively, I shouldn't use that word, is constantly back and forth because of cation exchange capacity. So when the proton pumps of the plants start putting protons in here, those protons will exchange on these cation exchange sites and put bases back into the system. Okay? And in essence, equilibrate that pH, buffer that pH. But in time, as I said before, in time, we're going to start running out of the bases that are here. And sooner or later, we're literally going to run out of this material because it's going to be weathered away. And in time, the proton concentration in these two sites are going to increase relative to the bases, and that's when the pHs start to drop. Okay, but this is the pool that controls that nutrient dynamics, and it controls the nutrient availability for plants. Cool? All right, so let's actually talk about this total acidity. It turns out, well, I mean, let's use this slide. It turns out if you think about managing the soils, we can only measure this. But if we look at this whole issue of this cation exchange capacity, we realize that if we want to manage for nutrients, we actually have to manage for this pH as well. It's not just this pH. Okay, so we really, when we think about management, we really have to think about the total acidity. And now this is what we call, this is called the coffee cooler analogy or visualization. What you want to look at here is you want to imagine all these different acidity pools, okay? And you think about the liquid in the container being that pool of acidity. Now it turns out that the smallest pool of acidity is this active pool. This pool is the smallest pool. It's extremely important, but it's the smallest pool. Wrong way. The size of the cooler, I'll use these two as an example, the size of the cooler represents how much acidity is in that exchangeable or salt replaceable as well as the residual or the structural. Okay? So imagine I have a soil which has a high cation exchange capacity versus a soil that has a low cation exchange capacity. Now, if I have a soil that has high exchange capacity, cation exchange capacity, and it has high base saturation. You guys remember what base saturation, right, is? Base saturation is the percentage of cation exchange capacity that's occupied by bases. All right, so here's my water cooler. Nice big water cooler here. It's got a CEC of 100, okay, and it's got a here's its active pool. Here's another water cooler. This one has a cation exchange capacity of 10. The active acidity pool is the same. Okay? Now base saturation. Let's imagine for whatever reason I have 100% base saturation. So I have in here 100 positively charged cations. They're attached through cation exchange capacity, right? In here I only have 10. Right? So here's my level of pH. Let's imagine this is high pH up here, so 12, and this is 6 down here. Okay? 12 and 6. Does that make sense? Okay, so this is in essence a scale of pH. I start adding protons to this system. What's going to happen to the active pH? It should start dropping, right? So my pH is going to start moving down in both scenarios. But this pool is going to start interacting with the cation exchange sites, right? It's going to start reacting with that exchangeable acidity pool. Now, if I added, this is not the way you do the math, but if I added 6 protons to drop this down, potentially 5 of those protons are going to react, and I now have 95% base sat over here because those bases are now coming out. Okay, and in the process, it's going to take my pH back up. Not to where it was, but it's going to take it back up. Now I do the same thing over here, right? I add 6 cations. No, I add 6 protons. I'm dropping the pH. Those protons go over here to the exchange sites. The bases get knocked off. Let's imagine I take 5 of them off. Okay? So I now have 5% base saturation here. I have 50% base saturation, which means I literally only have 5 bases left, right? Does that make sense? Go.
So the second example was originally it was 10 CEC and 100% base saturation.
100% base saturation. Okay, so both of these guys, when we started, had 100% base saturation. But this guy had a CEC of 100. This one had a CEC of 10. Right? In essence, I exchange 5 sites. I get 5 protons going on to the exchange sites and 5 coming off. Base saturation here is 95%. What's base saturation here? 50%. Okay? Those bases go back into the solution and they raise the pH back up a little bit, right? Question?
Is there a reason that only 5 out of the 6 protons react?
Well, it's exchange reactions, you know, so it's not going to be 100%. Okay? All right. That's what happens. Now, we're now at stage 2. Somewhere down the line, I decide, whatever reason, something happens, more protons go into solution. Right? And that's another 6 go in. Well, what's going to happen? The exact same thing's going to happen again. We're going to have this initial drop of pH. It's going to go a little bit lower this time. We're going to have the exchange. Okay? We're going to lose some bases. And this base saturation now goes down to 90. Right? We've lost 5. Same thing happens here. We have an initial drop. It drops a little bit lower. In this scenario, I put 6 in the system. Okay? We have 5 of them taking over. 1 of them doesn't. Okay? But the 5 bases that are here go into solution. Raise the pH back up. What's my base saturation now? 0. Does that make sense? So in essence, that exchange acidity is buffering, mediating, slowing down, moderating the pH change, right? Scenario number 2. Okay, now scenario number 3 comes along. Another incident. Okay, we add another 6 protons. Right? And here, 6 protons. Again, we have this drop. It goes down a little bit. We have an exchange. We're now at 85% base saturation. pH goes back up a little bit. It's a little bit lower than when it started. But this guy, what happens here? It doesn't have anything to exchange anymore. What's going to happen to the pH? It's going to drop, and it's going to stay down. Because I've just added 6 protons. And yeah, they're going to be exchanging, but they're going to be exchanging for other protons. Does that make sense to everybody? Okay. So with this graph, I've taken a number of steps here. In fact, I've basically presented half the lecture right in this graph. Okay? But the take-home point with this slide is you have to consider the entire acidity when you start thinking about basically managing. Because we can take this scenario and throw it the totally other way. What happens if I start adding bases? What's going to happen? Okay, imagine that I add 10 bases, 10 charges, 10 positive charges that are bases. Okay, in this scenario, you know, if it's not going to be 100%, but in this scenario, if I add 10, I'm going to basically put this right back up to 95%, base saturation to 95. I'm going to be raising the pH, and then it's going to drop back down, but it's not going to drop down as low as it was. Does that make sense? In this scenario, what's going to happen? If I add 10, and granted, we're not going to get 100% exchange, but if I add 10, we're now back up to 100% base saturation. My pH is going to bounce back up, right? But what happens again? This guy is back at 95. What happens if I add 5 bases again? And I'm going to bring this guy back up all the way to 100% base saturation. What's going to happen in this scenario, though? Where are those bases going? They can't go there. If they're not taken up by the plant, what's going to happen to them? They're going to be leached from the system. So not only have we talked about pH, we're also starting to talk about fertilizer. I need to know what my cation exchange capacity is. I need to know what my total pool of acidity is so that I can, in fact, manage for these management scenarios. Make sense? Cool beans? Questions? No? All right. All right. I use these, there's 2 slides here, and I use them as segues, and it's really the, sort of the segue, what is this relationship between pH and exchangeable cations? Okay? Because you got to think about this. What happens to the pH when, what happens to the CEC when pHs change? Now, I have a permanent charge associated with my cation exchange capacity, but I also have a pH-dependent charge, right? So that pool of base saturation is going to change based on the pH. right? In this scenario, we're basically assuming it was a permanent charge. But what happens if that CEC is not static? It changes based on what my pH is. And so right here, this graph basically looks at percent of maximum cation exchange capacity. Okay, this is how much you can hold. Okay, and this is soil pH. Okay, and this is a mineral soil, where this line right here represents, or I should say everything below that line basically represents permanent charge, and everything above that line basically represents pH-dependent charge. As the soil pH increases, my effective cation exchange capacity also increases, which means that pool is going to get larger. That exchangeable pool is going to get larger, right? As the pH goes up, as the pH goes down, that effective capacity is going to go down. Now, what happens when I start adding organic material in here? Organic material has a much, much larger pH-dependent charge associated with it. Here's the permanent charge right here, everything below that. Everything above that is pH-dependent. So as my pH increases, my effective cation exchange capacity increases, hugely affecting those pools. Okay, questions? All right. This is just a slide I threw in here to guys to remind us of what base saturation is. Base saturation is just that measurement of the cation exchange capacity that's occupied by bases, the percentage. All right. Let's skip this. Let's actually get to management. All right. These up at the top there is basically a scale that shows pHs, 5.5 and 8.5. So anything lower than 5.5, we're basically talking about, from an agronomic sense, anything that's below 5.5, we're usually talking about a scenario where we need to add lime. We need to bring the pH up because, remember, it's that active acidity that controls the bases. It's controlling the nutrient dynamics. Okay? So I want to basically have my pH between 5.5 and 8.5 because that turns out to be the maximum zone for nutrient availability. Now, if it's above that, I may in fact have to do some acidification because if I get up to these high pHs, I might start seeing micronutrient deficiencies. It's about nutrient management. Okay. So what are some of the things that we potentially can do? Well, soil pHs shift on this scale due to a number of things. Cropping in humid climates will slowly decrease the pH. Why? If I'm cropping, I'm harvesting, right? And if I'm harvesting, I'm taking the bases out of the system that the plant has harvested from the soil solution. Again, if I start taking bases away, what's going to have to happen to that relative concentration of protons to bases? The proton concentration is going to start increasing. And if I start removing bases, I no longer have the bases that can exchange on the cation exchange to, in essence, buffer the soil. So as a result, my pHs are going to start dropping. Okay, now the truth is, even if I don't crop and I let it sit there for a couple thousand years, my pHs are going to start dropping because those bases sooner or later are going to be leached out of the system. Okay, pH in arid climates actually increases due to the alkalinity in incoming water. Well, what's going on there? Because of evapotranspiration and the bases being left behind, when I do have water, groundwater moving through the system, or I use water in the system, they tend to have a lot of bases. Okay? So that water is in essence going to be bringing those bases into the soil solution. In arid environments, groundwater tends to have higher pHs, unlike rainwater, which tends to be acidic. Got it? All right. Which brings us into the issue of buffering the soil. We want to control the pH of these soils. We want to basically put it into a certain zone. Okay? And really, what we're talking about is reducing the extreme nature of these events, reducing the extreme changes of pH. We want to slow it down, and we want to basically contain it or maintain it in a certain zone of pH. Does that make sense? Okay. Now, there turns out to be basically 3 different types of buffering reactions. This is, I threw this in here, and I actually already did it over there. But this is the sort of the back to that analogy of the water cooler. This is a large CEC. This is a low CEC. Okay? This pipe right here basically represents the active pH. Okay? And you can certainly imagine, which happened over here, if I have a high CEC that I have 100% base saturation, as I start adding protons into the soil solution, the bases that are on the cation exchange capacities that are in base saturation will exchange, and they'll resist the pH change. Okay? In this scenario, I have a much smaller water cooler, which is the one on the right, the scenario on the right. And yes, it will resist pH change, but it has a limited capacity to resist those protons coming into the soil solution. Does that make sense? Yes? Go.
Unless it's the third acidity, is that referring to the residual or the CEC?
This is basically referring to both of them, but really it's talking about the CEC, the exchangeable acidity. Okay? Yeah, that makes sense? Okay, did anybody have a question about this? Because I want to take the next step, which is actually talking about buffering. Okay, now imagine we have 2 soils. Okay, and one soil we have this really big CEC, high base saturation. One is we have a small CEC, and it doesn't matter if we have high base saturation because we have a very small pool. Okay, if I have a really well buffered soil and I start adding soil protons basically to it, so this is the amount of protons down here, or the amount of bases that are added off of this neutral point, and this is the pH of the soil. These lines represent the pHs of those soils at certain concentrations of added or subtracted protons. Okay, so if I have a well-buffered soil here, high CEC, high base saturation, and I start adding protons to it, I'm going to have a very slow response pH-wise. Because for every proton I throw in there, I'm potentially going to get a base bouncing off, which means it's in a sense neutralizing that acid. It's a base cation, right? Now, if I have a soil that has this, the one on the right here, that has a very limited amount of buffering capacity, it has a small CEC, and while it may have 100% base saturation, it has a small amount of bases on those exchange sites in total. So for every one that I throw on, yeah, I'm going to get a bounce off, but I only can do it 10 times. And so as a result, I'm going to have this huge response. Okay, this black line here basically represents water, where I in fact have no buffering capacity. For every proton I add in, I basically get more acidity. I'm not going to get any kind of buffering reaction. And so as a result, the pH change is rather dramatic depending upon whether I'm adding bases or protons. Does that make sense, everybody? Questions? No? Go.
So you have 2 examples of buffers, like human compounds,
Yeah, well, but we're going to talk about that in a moment. Yeah, there are multiple kinds of buffers, and these buffers are working at different pHs. They're active at different ranges of pHs. Okay. Feel comfortable with this so far? So, because what I want to do now is I want to actually talk about what those buffering reactions are, what the things are that are in the soil that do this all the time. Buffering at the middle. Buffering in the middle is basically cation exchange. Okay, I start throwing protons in. Those protons are going to go in the soil solution. Those protons that are in the soil solution are going to exchange with the cation exchange sites, and in essence, I'm going to have a buffering reaction. Okay, the protons are going to be neutralized by the bases coming off the cation exchange sites. Okay, this is what we've been talking about for a while, but basically, here's my aluminum with hydroxide. That proton hits the soil solution. That proton reacts with the cation exchange site. It's basically held, and it's neutralized. Okay. You can think of all these cation, this is the clay micelles, but basically we're looking at the active pool here, but this cation exchange is going on here. If it's a proton reacting here, I'm buffering it. And it generally happens in this 4.5 to 7.5 range. Okay? Buffering at the edges, basically we're looking at buffering at higher than 7.5 and buffering at lower than 4.5. There's 2 different reactions that are associated with them. At the buffering at the high, it's basically a calcium carbonate reaction. Okay? Now remember this reaction because this is the reaction of water. And we're going to take a first step. CO2 with water basically makes carbonic acid. It's a weak acid, so only one of the protons dissociate. It's a strong base. Okay? But this strong base can react with calcium. Okay? Basically, the calcium will react with this base. Okay? And as a result, I'm going to be putting in 2 protons into the soil solution. So when the pH starts getting higher, I'm going to start seeing more calcium in the soil solution. This calcium will react with this carbonate base, and it will knock that proton off. In essence, neutralizing the raising of the pH. Because it's going to make a calcium carbonate, and as a result, I'm going to get these 2 protons. Does that make sense? The byproduct of it is water and CO2. We started with water and CO2, and we end with water and CO2. No? Kind of? All right. Let's take a step back and let's think about where this comes into the system. Okay. I have a high pH. My soil has a high pH. What does that mean? Well, it means I probably have high base saturation, right? So my colloid, and we'll stop after this and I'm going to kick you guys out. Okay, my colloid on the exchange sites probably has a lot of calcium. High pH, right? I have a rainwater scenario, H2O plus CO2, right? Makes carbonic acid, H plus HCO3, right? This proton goes into soil solution. What happens to it? Potentially, it's going to be reacting. Okay? Means that calcium is now going into soil solution. And even if it doesn't, because of the high base saturation in these systems, I probably already have calcium in soil solution. So what's going to happen with this carbonate? This calcium, strong reaction with this base, basically making calcium carbonate. As a result, I'm releasing 2 protons. Those 2 protons are going to go into the soil solution. What are they going to do to the soil solution's pH? They're going to lower it. So in essence, they're resisting that pH change. They're making it more acidic. Okay, because of this carbonate reaction with the calcium forming calcium carbonate. It's a buffering reaction. It's resisting the pH change. Make sense? Questions? This happens at the high-end pH range because I need to have the presence of this, or potentially magnesium, but we'll talk about that at a different time. Okay? Feel good? All right, guys, have an awesome weekend. Sorry about the rain. Be free.